Hello students, today I am going to talk about the topic receptor concept. Here I will discuss one receptor in detail, and the part 2 receptor concept. In the next video I will discuss rest of the receptors. As we have already learned about the pharmacodynamics, so everything what you have learned in pharmacodynamics going to be useful to understand the receptor concept. As I told you in beginning as well, the receptor concept is very important topic, because without understanding this you cannot get the confidence in the pharmacology. And once you understand this whole receptor concept, you will be able to explain the mechanisms of the drugs and their other minutes about this. So let's begin the receptor concept. Already I have given you the definition of receptor. Here I just want to revise for you. A very simple definition I have kept. So receptors are molecular substances or macromolecules in tissues that combine chemically with the drug. So two important points here, words here, molecular substances, and they are, you can say, the macromolecules. Drugs have a considerable degree of selectivity in their actions. You know very well that every drug has a one specific action, and they want to produce the specific action what for you have made them. So that specificity comes by the selectivity. It follows that the receptor with which they interact must be equally unique, right? So they should bind with the receptors which are uniquely acting for them. For example, one example I have taken here as the end plate region of a skeletal muscle fiber and acetylcholine. So you see the receptors which are available on the skeletal muscle end plate, and there the acetylcholine binds with those receptors. So what happened? You see here, acetylcholine receptor, when binds with the acetylcholine, sodium influx happens. Sodium influx gives the depolarization that will help to generate the action potential, and this action potential will increase the free calcium level, and ultimately you will get the effect of contraction. So like that, a receptor binds with its drug binds with this receptor, and by chain of sequence it will give you the final effect. Before understanding the receptors and their working physiology, first we need to understand the second messenger system, right? Already I have spoken about this in the previous video. So second messenger system means, for example, you can consider your drug is a first messenger. Means drug has the information that what to do, what for you have taken the drug. Suppose that you have taken paracetamol, you want to relieve your pain or you want to relieve your fever. That's what the message paracetamol have. So once paracetamol goes into your body, or you can consider any other drug, it has the information, and that information has to pass on to someone to produce the effect. So that another molecule which responsible to convey this message further will be called as a second messenger. They all are the different proteins. Sometimes the second messenger will again convey it to some other protein. That protein may be a third messenger. So like that, these proteins are always available. There are certain receptors which are using these second messengers to produce their effect. Some receptors do not use these second messengers. Key factors in many of these second messenger systems are proteins termed G proteins. So most of the cases, this in the second messenger system, they have one very important protein which we call it as G protein, and the G word stands for guanine nucleotide binding proteins. G proteins have the capacity to bind guanine triphosphate and hydrolyze it to guanine diphosphate, right? So always G proteins are hydrolyzing the GTP to GDP, right? That's what they are doing, and by that they are generating some signal and the further events. By this slide you can understand how this second messenger system actually works. You see in this first diagram, you can see here neurotransmitter comes, releases and binds on the receptor here. Once it binds to the receptor, this receptor get activated, and the activated receptor has activated the another protein. This protein is a, for example here it's a CAMP, right? And this CMP protein is actually acting as a second messenger here. So the message has been this hormone has activated this receptor has activated the second messenger, and the second messenger will further activates the another protein which we call it as here, for example, CREB protein. This CREB protein will ultimately reach to the nucleus, and it will activates the gene, that a particular gene. Once that particular gene get activated, you know very well what happens next. The gene will go into the protein synthesis process, and by that that gene will produce certain kind of proteins, and these proteins will produce the desired effect what for the hormone released. Okay, so this way the second messengers are acting. That has been shown into this blocks as well. So agonist activates membrane bound receptor, G protein is activated and produces effector. So what happens here, the receptor which is available in this particular example, that receptor is having one additional protein attached with the receptor, that additional protein is the G protein. So once the hormone or a drug binds to the receptor will activate that particular protein which is a G protein, right? And then G protein will activate the second messenger. Then the second messenger will activate the further other proteins and to perform the action. So likewise the second messenger system works here. This is another very important or very good diagram to explain this same thing. Here you can see at the first place it is written hormone. So hormone or can be a drug or can be other chemical or any factor. When it is binding to a receptor, we call this as a first messenger. So that is a first messenger because that only have a first message what effect has to be produced. Okay. Now you can see this blue color structure. This is your G protein, and this G protein is right now is having a GDP, which is later once the receptor get activated, this GDP will become GTP, right? So this GTP means the activation. This G protein now got activated. This G protein now got activated. So this is an activated G protein. Now this activated G protein will go and bind with another enzyme here, right? Yes, another enzyme. So this enzyme, you know, these enzymes are always catalyzing the reaction or the process. So here also the enzyme will do the same, and the enzyme is here is the adenylyl cyclase, right, or adenylyl cyclase enzyme. Now what this enzyme will do? This enzyme will hydrolyze. Hydrolyze what? It will hydrolyze the ATP. It will hydrolyze the ATP and this ATP will convert into ADP, and then ADP will convert into AMP, right. So, this is the AMP here, and this AMP will again adjust its structure and it will become cyclic in nature, cyclic in structure. So, we call it as a cyclic AMP, right. So, now what this adenylyl cyclase has done, it has hydrolyzes the ATP and converted into CAMP. Now, this CAMP, what it will do, this CAMP is actually your second messenger, right. This CAMP is your second messenger, right. And now this second messenger will convey its message again, and by that it will activate the another protein which we call it as protein kinase, right. Now, this protein kinase, once it activates, it will perform various cellular functions, right. Maybe it will activate enzymes, stimulate cellular secretions, opens ion channel or something. So, it will do various processes here, right. So, the same CAMP second messenger has been used by various hormones, for example, epinephrine, glucagon, ACTH, PTH, FSH, LH, TSH, calcitonin and many more. Okay. So, this is the way it happens. Another diagram is here because I have given it to make you understand one thing, that every time the enzyme which is being activated previous in the previous slide was adenylyl cyclase. So, it is not the same that every time adenylyl cyclase will only activate, it depends that maybe a different enzyme will activate. So, here in this particular case you can find another enzyme which has been activated, that is a phospholipase. So, always remember, whenever the enzyme changes, it will change its substrate also. Okay. So, in the previous case the substrate was ATP, right. Now, in this case the substrate is PIP2. Okay. Now, this phospholipase will hydrolyze PIP2 and will convert into two molecules. One is DAG, you can see here, one is DAG and another one is IP3. Okay. Now, the phospholipase has hydrolyzed PIP2 and made DAG and IP3. Now, these DAG, diacyl glycerol, and IP3 both are acting as a second messenger, right. Both are acting as a second messenger, means you can say here in this case two second messengers are generated and both are doing different actions here, right. And after doing these different actions, either they will activate the certain protein kinases, as the DAG will do that, DAG will activate the protein kinases, and then it will act on the calcium release, right, and then it will perform certain actions. Same way IP3 will also do this, the different actions. So, different hormones are there which are using this, for example, TRH, ADH, LHRH, oxytocin, right, catecholamines, they all are using this kind of pathway. Understood? So, this way the second messenger system works. These other things I will tell you in detail in coming slides. So, now you understood what the receptors and what the second messenger system is there. Now, we will see the something about the chemistry of the drug receptor binding. Already you have seen in the previous presentation as well that the binding between the drug and the receptor needs various kind of bonding system. So, here we have the various bonds: hydrogen bond, ionic bonds, van der Waals forces, covalent bonds, and like that these bonds are there. You can see covalent is the highly stable bond, strengthful bond, and the van der Waals is the lowest. Whenever the drug binds with the receptor, it is a cyclic process that it binds and then moves out from that, right. So, it is having the association and then it is also dissociates from there. So, the association and dissociation is a kind of cyclic processes which are keep on happening, and this association which we have said in the previous presentation, affinity. So, that is what the ability of a drug to bind with the receptor or associate with the receptor is the affinity what we have said. Now, before going into the individual receptors, let me tell you about the general, which basis actually we have categorized the various receptors. So, what are these bases of the classification of receptors are? One is the pharmacological criteria or the pharmacological basis. So, this is a very common and old method of classifying the receptors, right? And if, for example, you can say uh, muscarinic and nicotinic cholinergic receptors, alpha and beta adrenergic receptors, H1 and H2 histaminergic receptors, etc. They all have been divided on the basis of the pharmacological criteria. Usually here their relative potencies has been checked, right? On the basis of their relative potencies, the division can be done. Another is the tissue distribution. This method is usually applied while categorizing the subtype of the receptors, right? So, for example, here beta adrenergic receptors, if it is present in heart, we call it beta 1. When it is present in bronchus, it is called beta 2. So, beta is the receptor, but as per its different location, we call it as an beta 1 and beta 2. So, this classification happened on the basis of their tissue distribution, right? One is present on cardiac tissue, another is present on the lungs tissue. Third is the ligand binding, right? Which ligand is binding on them, that is also another basis of doing it, doing the classification. In this, 5-HT receptors are one of the example by which like receptors been classified, that which ligand is binding on them, right? Or what is the agonist which is binding on them. On that basis, they have been classified. Next is the transducer pathway. This is a very important classification of the receptors. Here what happens, you know what is the purpose of a drug. When you have taken a drug, you need to get the effect of the drug. So, from the time when the drug binds with the receptor up to the time when you get the response, that whole process is the transducer pathway, right? That is the whole process, the transducer pathway, by which ways you get the effect, that is called transducer pathway. That is another method of classifying the receptors. For example, muscarinic cholinergic receptor, which has been denoted by M here. Muscarinic cholinergic receptor acts through G proteins, while nicotinic cholinergic receptor gets influx of sodium ions. So, like that, there's a difference in their mechanism. Same way you can see in case of alpha adrenergic receptor, acts via IP3DAG pathway and by decreasing CAMP, while beta adrenergic receptor increases CAMP. Okay, so the different method of action. GABA A receptor is ligand gated ion channel, while GABA B receptor increases potassium conductance through a G protein. So, likewise, various different receptors having the different transducer mechanism, transducer pathway. So, on that basis, it has been divided into them in different categories. Okay. The last method of classification is the molecular cloning. This is the recent time this molecular cloning has been started to categorize the receptors into various different types and subtypes, usually subtypes. And this has been done by by the finding out the sequence homology, right? And that sequence homology, or you can say the DNA splicing or RNA splicing, how it happens. On that basis, these different type of receptors have been formed. So, they are having difference in their amino acid sequence as well as their three-dimensional structures, right? Like that, these classifications have been done. This slide will give you one general information about these 5 receptors, where the receptor number 2 and 3 are almost of the same category receptors. And here you can say first one is the nuclear receptor, nuclear hormone receptor, which you also call as steroid receptors. Second is the enzymatic receptors, and third is the receptor linked to kinase subunit. So both are having almost similarity. Fourth one is the ligand gated ion channels, and fifth one is the G protein coupled receptors, right? And below it has been given that what is their how much time they take to produce the effect. In the next slide it is more clear to you. Here you can see the 1, 2, 3, 4 structures have been shown. Ligand, first one is depicting the ligand gated ion channel, which they also called as ionotropic receptors. Second segment shows the G protein coupled receptors, which we also called them as a metabotropic receptors. Third shows the kinase linked receptors, and fourth shows the nuclear receptors. So you can see the first one, ligand gated ion channel, which is acting through ion channels, and once the ion get bind with the channel get open, and then hyperpolarization or depolarization happens, and then the cellular effect produced, and usual time needed for it is in milliseconds, and examples are nicotinic and acetylcholine receptors. In the second category, it's a little another extra part is added, that is second messenger system, right? In the first case, in the first ligand gated ion channel, there is no second messenger needed, but in the G protein coupled receptor there is one second messenger needed, and that second messenger, when it get activated and it further activates other proteins, other or phosphorylates other proteins, then the cellular effects produce, various cellular effects produces. The time needed for it in seconds, examples are muscarinic and ACH receptors. Third category is the kinase linked receptors. Here also one enzyme, additional enzyme is added with the receptor, they are called kinases, and these they get activate, and then they do the gene transcription, and they modify the protein synthesis as well, and then by that they produce the cellular effects. So examples are cytokine receptors, they are called cytokine receptors as well, and the time needs little more, that is in hours. Next is the nuclear receptor, where the receptor is not present on the membrane, but it is present inside the membrane or inside the cell, inside the nucleus, right? So they are called nuclear receptors. They also having the capability to change the gene transcription and affect the protein synthesis, and by that they produce the various cellular effects. That also needs hours to produce its effect. Various hormones are using this method, for example estrogen receptors. So I hope you understood a broad category of the receptors and their divisions. In this screenshot you can see there are 4 different receptors, their subtypes and indigenous ligand what they use it. First one is the ligand gated ion channel, LGIC. For example, acetylcholine, GABA, glutamate, serotonin, right? They all are using these receptors. Their subtypes are nicotinic, ANC, glutamate has NMDA, kainate, AMPA, serotonin 5-HT3 type. Indigenous ligands are using this, acetylcholine, GABA, glutamate or aspartate, serotonin, right? Same way for second one is the G protein coupled receptors. There are also their type, subtype and indigenous ligand, RTKs, receptor tyrosine kinase and nuclear hormone receptors. So all 4 receptors with their subtypes and indigenous ligand. Please go through patiently so you can remember this. So we'll move now further, and we'll see here if we talk about the neurotransmitter receptors, means the receptors which are needed by the neurotransmitters to produce their effect. They are categorized in 2 categories. One is called ionotropic receptors, and these are ion channels, as I have given you, because ionotropic word they are ion, their ligand is ion, are ion channels and found in fast chemical synapses. Acetylcholine, glycine, glutamate, GABA all acts on the transmitter gated ion channels, mediating excitatory or inhibitory signaling, right? So that's what, either they will produce the excitatory signals or they produce the inhibitory signals, right? Metabotropic receptors, for example the GPCR, that binds to all other neurotransmitters along with acetylcholine, glutamate and GABA. and signaling tend to be far slower and more complex than that at ionotropic receptors. So always keep in mind the ionotropic receptors are very fast, but the metabotropic receptors requires having more additional processes inside. That's why they need more time. Now, when a drug binds with a receptor, how many type the drug can bind to a receptor, right? So there are 6 types have been divided. First is a transmembrane ion channel. Transmembrane ion channel means a drug binds to the ion channels which are present transmembrane in the transmembrane. Transmembrane one word means across the membrane, right? So they are present across the membrane. Those the ion channels which are present across the membrane, there is drug binds with them. Then it's called transmembrane ion channels. Second is transmembrane receptor coupled to intracellular G protein. Another type of interaction is drug interacts to those kind of protein receptors which are having one additional protein which we call G protein. Third is a transmembrane receptor with enzymatic cytosolic domain. Means another interaction method is the drug binding to that kind of receptor where that receptor have one part of the receptor itself is an enzyme, right? It will not activate other enzyme. The one part of the receptor which is a cytosolic domain will have the enzymatic capability. Fourth is the transmembrane receptor including enzymes, transcription regulators and structural proteins. This is another type of interaction where it can interact with directly with the enzyme, transcription regulators or maybe the structural proteins. Next is the extracellular enzyme. So some drugs are acting through extracellular enzymes as well. For example, ACE and acetylcholinesterases. And the last interaction type is the cell surface adhesion receptors. Some drugs binding on the cell surface adhesion receptors, receptors which are adhered on the cell surface, right? For example, integrins. So like that, these are the 6 various type of the drug receptor interactions. Now I want to discuss the transducer mechanisms. From here, now you will start building your platform to understand that how to define the mechanism of action of any drug, right? So here this transducer mechanism, if you understand this properly, you will be able to write the mechanism of action of all the drugs, right? Because this will give you the general basis of writing that or understanding that. So the receptor response is highly complex multi-step process. Whenever we talk about the transducer mechanism or drug's effect, from the time of binding up to the time of producing effect, it's a multi-step, a complex process. Maybe the diagram which I will show you, it looks simple, but it's not that simple. Nothing is simple there, right? Because it requires a multi-step process. Right here we we'll short the process steps and we'll show you in a simple form. These mechanisms explain the translation of receptor activation into the functional response. So ultimately what these mechanisms are giving you, from the time of receptor activation up to the time of functional response of the drug. Now, when we talk about the transducer mechanisms, we have to see how many type of transducer mechanisms are there. And it's very nice that you have only 4 type of receptor mechanisms or transducer mechanisms. It means all the drugs in the world which you have can have only these 4 type of transducer mechanisms. So it's so simple for you. If you remember only one thing, that the particular drug, only you remember that this particular drug acts by the mechanism number 2, then that's all you know. And now you know which is the mechanism number 2, then you can explain it very nicely. So I'll tell you these 4 major categories. After this video and the next video, I want you all to have these transducer mechanisms in your fingertips. Okay. So first transducer mechanism is G protein coupled receptors. GPCR. They are also called as transmembrane receptors linked with G protein. Okay. Receptor with intrinsic ion channel. Transmembrane, you should be very clear about that. What do you mean by the transmembrane? Suppose that this is a membrane. So the receptor which is present like this. Okay, this is a transmembrane through or the across the membrane, right? So in the first case, if this is a G protein coupled receptor, then this is a receptor, and in the transmembrane level it has one additional protein. That's what it is called, coupled. Now this receptor is coupled with this G protein. Okay, coupled with this G protein, so that become G protein coupled. Coupled means linked protein, right? Second is the receptors with intrinsic ion channel. They are also called as ligand gated ion channels. So what is going to happen here? One receptor is there which has a one ion channel in that. Third is the enzymatic receptors. They are also called as ligand regulated transmembrane receptors with enzymatic cytosolic domain, LGICS. This category, enzymatic receptors, are having two distinct mechanisms or two distinct method of working, right? In this one, which we call it as an intrinsic enzyme receptors. Second, we call it as JAK-STAT kinase binding receptor. They are also called cytokine receptors. You can also call them receptor tyrosine kinases, RTK, right? Last one is also very important, intracellular receptors. These intracellular receptors, also called as the receptor regulating gene expression. Some books will write like that, receptor regulating gene expression. Some books say nuclear receptors, cytoplasmic receptors, or intracellular receptor for lipid soluble agents. So remember, these are the four transducer mechanisms which comes under the receptor category, where you will have the mechanism of their action. Now I'll begin with the first and very important receptor, which we call the G protein coupled receptor. Right, in the left, what the kind one structure is being shown. This structure is a G protein coupled receptor structure, right? And as I told you, they are across the membrane or the transmembrane. They are, right. So we will discuss about the G protein coupled receptor. And here, when we talk about the G protein coupled receptors, we have to see the structure of the receptor, as well as we will see the mechanism of the receptor and how this receptor actually produces its effect. So the G protein coupled receptors are the most abundant class of the receptors, right? They have a very huge family of receptors here. And you can see in the coming slides that they are exposed at the extracellular surface of the cell membrane, and they also have one intracellular region because they are transmembrane. So they have the two regions. One is present at extracellular part, another is present on the intracellular part. And in the intracellular part, they are activating a very unique protein or signaling molecule, which we call it as a G protein. You can see later. So here the large number of G protein isoforms are also being identified, right? And they also have their unique effects on their targets. And these proteins are distributed among various genes, means various genes are producing these various isoforms of the G protein. So in this way, this GPCR can be grouped into 6 classes based on the sequence homology and the functional similarity. You can see the class A, B, C, D, E, and F. There is also called as class A 1, 2, 3, 4, 5, 6. So if you see the class 1 have 662 genes, means it distributed to 662 genes, class B 15 genes, class C maybe 22 genes, and class D, E, F comes under the others, which are having 92 genes, right? That way it is been distributed. And if you see the class 1, which is called as the rhodopsin like class, here they are two type of in that also the two specifications are there. One is endogenous ligand or orphan type, they are having 271 genes, and olfactory and pheromone, that is 391 gene. Like that it is been categorized. And class B, which is secretin receptor family, class C metabotropic or glutamate pheromone, class D fungal mating pheromone receptors, and like that different classes are having a different type of receptors which are there, right? In this classification, later you will understand that the rhodopsin receptors, uh, gasteucin receptors, olfactory receptors, serotonin receptors, dopamine receptors, GABA receptors, glutamate receptors, chemokine receptors, histamine, sympathetic, parasympathetic receptors, they all comes under the GPCR category. Okay, so that's why it is one of the very, very big family of the receptors. So before going into the further detail, let's see the targets for G protein. Adenylyl cyclase. So this G protein which is present in this particular receptor has the following targets. It will activate these targets, right? Adenylyl cyclase. So the enzyme responsible for CMP formation, or phospholipase C, the enzyme responsible for inositol phosphate or diacylglycerol formation. Ion channels, it has a target ion channels as well, particularly calcium and potassium channels. It also has a target for Rho A or the Rho kinase, a system that controls the activity of many signaling pathways, controlling cell growth and proliferation, smooth muscle contraction, etc. So likewise, this particular G protein targets these all category of molecules. Now when we'll see this particular structure, you can understand here that this G protein, already I have told you that it is named so because it's having the guanine nucleotide attached with it, right? So G protein coupled signaling mechanism involved in many important processes as in vision, olfaction and neurotransmission, right? So if I tell you, you can see the things, you can, smell the things because of G protein coupled receptors, so it's true. So these receptors you can see having a very specific structure where you can find they are having 7 alpha helical membrane spanning hydrophobic amino acid segment, right? So here if you see, this is a protein structure where they have 1 2 3 4 5 6 7, like that, 7 alpha because they are alpha protein, helical because they are helix structure, so they are helix in structure. These all terms what I am speaking are very, very important, right? Very, very, very important terms. So 7 alpha helical membrane spanning means entering into that, so spanning hydrophobic amino acid segments. Okay, and if you see in this structure clearly, you can find there are 3 extracellular loop number 1 2 3, and there are 3 intracellular loop number 1 2 3. Okay, likewise they are having one free end and the extracellular side and one free end at the intracellular side. So like that the structure has been structured right here. So I hope you are clear about the structure here. And now in this particular structure, if you want to see where the agonist binding site is, so the agonist binding site is located at extracellular domain of the helix, somewhere at the extracellular site. This is your ligand binding domain, right? So in this particular site, the ligand is actually binding. Another recognition site formed by the cytosolic segment. Now come to this cytosolic. This is whole the cytosolic part or the intracellular part. In this, if you see, you can understand that non-covalently linked one G protein is available. That consists, you see, this is the G protein. So this is the G protein which is available. In the other structures, you will understand this thing in a very better way, that this G protein is having 3 subunits. This G protein which is here is having 3 subunits in this, right? These subunits are called as alpha, beta and gamma. So they are having alpha, beta, gamma, 3 subunits present in this. So in this also you will understand that these alpha, beta, gamma have the different roles to play in the functioning of the G protein. So I hope you understood what the structure looks like, right? The inside free end is the carboxylic end, right? And the outside free end is having the nitro terminus. So like that the structure look like. So these are sometimes called metabotropic receptors, already I told you, and the structure comprise 7 membrane spanning alpha helix, often linked as dimeric structure, okay, and are having alpha, beta, gamma units in the G protein structure. And this I'll show you in this diagram here. You can understand. You see the first diagram which has been showing the G protein. You see here. These are the receptor, and these 3 are the G protein. 3 means they are subunit. They are looking one, but they are actually having 3 sections inside. So one is called alpha, one is called beta, another is called gamma. So whenever the activation of receptor happens, right, when the ligand bind to the receptor and the receptor get activated, the alpha is the one which get activated, right, mainly. So the GDP which is present with the beta has been donated to alpha, and now the alpha will become active and will convert this GDP into GTP. Now it will having a GTP. You can see here. Once the alpha gets GTP, you can say alpha activated now, right? So here it gets separated. This alpha gets separated from this beta and gamma, right? And this alpha is separated with beta and gamma, and then it will perform the next step that I will tell you later. Here also you can see this way. You can see here, this is your GPCR receptor, this is your ligand, and here the alpha, beta, gamma unit with GDP is present on the lower end of the structure, right? But right now this is inactive. When this GDP converts into GTP, then the activation happens, and that because of that activation, now you see here alpha, beta, gamma with GTP, now activated one, okay. Once it is activated, the alpha GTP is get separated. You can see here the alpha GTP gets separated, and beta, gamma together get separated from alpha. Now this alpha, which is an active unit, because of the GTP, it will, here is an effector protein. You see here is an effector protein. This is an effector protein. So this G alpha will activate this protein, right? This protein can be your enzyme, this protein can be any other factor. Most of the cases this protein is an enzyme, right? So this G alpha, GTP alpha will activate the effector protein, okay. Here I have shown you one animation. You can see that one ligand is coming, binding to GPCR. GPCR, the G protein get activated, and this G protein get activated. The alpha and beta gamma units get separated. You see the purple and green are the beta and gamma, and orange is the alpha one. And this alpha GTP has activated the enzyme adenylyl cyclase at the next. Okay, once this adenylyl cyclase got activated, this adenylyl cyclase will do further action. What it will do? It will hydrolyze the ATP, right? As I have shown you before, this adenylyl cyclase will hydrolyze ATP and will form AMP. AMP, and this AMP will again recycle, a structural modification happen, and it will become cyclic AMP. Like that, and this cyclic AMP will be your second messenger. I hope you understood. This is also one another animation which can help you out to understand the same way, talking the same thing. So you understood very well the active alpha subunit carrying GTP, which you call it as alpha GTP, dissociates from other two subunits beta and gamma and diffuses along the inner leaflets of the plasma membrane. You see, everything is happening on the inner side of the plasma membrane to interact with a number of different effectors. Okay, so here number of different effectors means maybe adenylyl cyclase, maybe phospholipase C, or maybe any other one. So this is the structure, gives you clear idea how the GPCR structure look like with the 7 alpha proteins, and below you can see the alpha beta gamma units, and the effector can be the enzyme or can be any other protein. So that's what the effectors get activated when the alpha get activated by the GTP. Here the sequential process has been shown. You can see by from the point 1 where the receptor, this is a very good diagram to tell you that how they are transmembrane, right? They are three loops of the structure receptors are outside of the membrane, and three loops are inside of the membrane, right? So it gives you a very clear cut idea. When the drug binds with the outer loop, the receptor get activated and it will activates the G protein, which is having alpha beta gamma unit. You can see alpha subunit releases GDP, binds GTP to activate. Once the alpha get activates, it will separate it from the beta and gamma. You see the beta and gamma has gone this way, and the alpha has activated one, came and bind on the effector. This is the effector part, right? Here, and you can see the separated subunits beta and gamma, they subunit activates or inhibit effectors. So they are also having some effect on some effectors, right? There they will do their activation or the suppression, and here the G alpha, which is actually targeted, is going to do the targeted uh, what called the activator to or the effector to activate or depress, right? So alpha subunit hydrolyzes bound GTP and becomes inactive. Now once alpha has activated the effector, immediately the GTP will again convert into GDP, right? And means alpha is now deactivated, and here beta and gamma has done their job. Now the beta gamma and the alpha, deactivated alpha, will come back again and get united together. Here you can see they get united together, right? So subunits combine to form inactive G protein, and then this combined form will again go and attach here. It's a regular process. So one cycle of the receptor activation and deactivation completes. Likewise, the G proteins are always acting. As I told you that this is not a very simple process, it's a multi-step process. So you can see Here various options has been shown. For example, the G protein have various agonists, right? Biogenic amines, amino acid anions, lipids, peptides and proteins and others. They can all bind with the receptor and activate it. Once the receptor get activated, the alpha, beta, gamma. So here alpha, there are various alphas are there, right? You can say alpha S is there, alpha I is there, alpha Q is there, alpha 12 is there. So there are various type of alphas are there, because and these different alphas are doing the different activations, right? They, because you know the alpha, activated alpha will activate the effector. So if the different alpha is there, then it will activate a different effector, right? That likewise the effectors will be changed, the activated effectors will be changed, right? And these different effectors ultimately will have the, by the multi-step process, they can have even the change in the genetics level. Okay. Here you can see, as I told you, that how this CAMP, which is this ATP, which is converted into hydrolyzes into AMP, and this AMP structure get into the cyclic form here. Because of this cyclic form, we call it as a cyclic AMP. So activation and inhibition, how it is happening, I already explained you. This is another way to explain it. This is the activation happening. You can see this is the activation, this side inhibition, right? Depends on which hormone or which ligand is binding. If ligand is activator, it will activate the process. If ligand is inhibitor, it will inhibit the process, right? Inhibit what? It will inhibit adenylyl cyclase activation, and here it will activate what? Adenylyl cyclase. Okay, so both depends on which ligand is binding. That ligand either will activate or inhibit the adenylyl cyclase, and in that basis, if it is inhibited, the no further activation will be happening, no further process will be moving on, and if it is activated, the further process will start. So like that, this process gives the activation and inhibition, depends on which alpha has been activated and which effector, and then what effect on the effector is going to produce. Another thing is one word we call it as an amplification. Amplification means what? Because there will be one receptor and one hormone molecule. So one hormone molecule bind with one receptor. So that cannot produce that much effect, which is an ultimate effect of the hormone, right? So it means for producing the required intensity of effect from a hormone, you need to amplify the effect, right? You can see here clearly one epinephrine molecule activates 3 adenylyl cyclase. These 1 adenylyl cyclase activates 2 CAMP, right? So it means 3 adenylyl cyclase, how many they have activated? Means one adenylyl cyclase activating 1, 2, 3, 4, 5, maybe 5 CMP molecules. Each one CMP molecules activates one protein kinase, so the number of protein kinases are also increased, right? And the one protein kinase is activating number of activated enzymes, right? So this is what is happening, and one activated enzyme will produce the various products. So this way the amplification process moves on, and ultimately we achieve the desired intensity of the effect. Okay. Here is a very important diagram to understand one thing. This how the different second messengers have been activated by the G protein. You see the receptors are there which activated the G protein. So target enzyme may be guanylate cyclase, may be adenylate cyclase, may be phospholipase C, right? These three may be your target enzyme. And these target enzymes are activating different second messengers. For example, CGMP, maybe CAMP, maybe IP3, maybe DAG, maybe some amino acids. These different second messengers may activate different protein kinases, maybe PKG, maybe PKA, maybe PKC, right? And these protein kinases will produce different, they activate different effectors, right? These effectors may be enzymes, maybe transport, maybe proteins, transport proteins, maybe contractile proteins, maybe ion channels, right? So this is the way the different different agonists are producing the different effects. So before moving further, I would like to tell you that the different forms which you have seen in the GPCR, alpha forms, and because of these different alpha forms, sometimes you can call it as an G alpha, GS alpha, or you can GI alpha, or sometimes you are not pronouncing alpha, but you are saying GS, GI, GO, GT, GQ, G12 by 13. So like that, it's all, its meaning is the different alpha protein which is going to activate, or you can say in common term different G protein. So one G protein which you call it as GS, S word indicates stimulation. So if the GS has been activated, it means stimulation is going to happen. If GI is activated, then it means some inhibition is going to happen. Getting my point? So what is happening here? The G proteins having a different type of, but actually what is different in that? Alpha one. So alpha is different, right? So there are various type of GI, GQ, GO. So they are producing different effect. For example, if I give you one example, for the muscarinic receptors, how many alpha can be activated by that? GI, GO and GQ. In the muscarinic receptor itself, there are three type of receptors, M1, M2 and M3. So in that, GI and GO can be activated by M2. GQ can be activated by M1 and M3. So like that, likewise, these different alphas get activated by different receptors and the different agonists. Here, before moving to further into the mechanism, here one point I would like to make it clear on this particular structure. The beta gamma which has been moved away, they are not also simply wasted, they are also having some role. So other than alpha subunit family, there are 7 beta and 12 gamma subunits are also there. As I told you, alphas are different, GS, GI, GO, GQ. So same way, beta and gamma are also present in many different forms, right? So here there are 7 type of betas are there and 12 type of gamma subunits are also there, right? So these other than alpha, beta gamma are released from the alpha subunit by GTP binding. Beta gamma also regulates some effectors such as ion channels and enzymes. For example, adenylyl cyclase B and phospholipase B can be activated by beta and gamma. Beta and gamma also activates muscarinic potassium channels in cardiac and neuronal cells, and is an important inhibitor of L and N type of calcium channels in neurons. One of the major role of G proteins is to activate the production of second messengers, right, that you have already seen. So examples of the second messengers have also you seen CAMP, calcium, potassium ions, inositol triphosphate, diacylglycerol and all, they all are playing this kind of role. Okay. So now you have understood the basics of the GPCR. You understood very well how the GPCR structure look like and how commonly this structure changes when the agonist binds on it, right? Now by this understanding we will move on to, there are three different mechanisms what the GPCR produces. So these are the number 1 called as adenylyl cyclase or CAMP pathway. Second, phospholipase C or IP3DAG pathway. Third is the channel regulation. In the channel regulation, there is no need of any second messenger, but in the first two, there will be a second messenger. Okay, and you see their name itself, by name itself, you can understand in the first one, the enzyme is activated is the adenylyl cyclase. In the second one, the enzyme is activated is phospholipase. In the first one, the second messenger is CAMP. In the second one, the second messenger is IP3 and DAG. Like that, by name itself, you can understand some of its information. Now, we will move on to the first one, that is called the adenylyl cyclase and CAMP pathway. Let us keep this GPCR pathways and the mechanism of receptor action for the second video. In this video, I will talk up to this point itself. So these are the references which I have used for making this video. Most of the diagrams what I have taken, it is from the Rang and Dale, the first book, as well as from the KD Tripathi, and I have used content from the Kadzang as well. So it's a very well balanced content taken from all these books, right? Thank you very much for understanding the receptor concept part 1 and especially the structures of the GPCR. And in the second part, I will discuss about the various pathways or the mechanism of action by the GPCR and other receptors. Thank you.